Hello everyone, this is Dr. Satish S. Bornare, Department of Genetics and Plant Breeding, Kikewak College of Agriculture, Nashik. Today we are going to learn the topic breeding methods for cross-pollinated crops. So friends, these are the breeding methods which were utilized for improvement of the cross-pollinated crop, in which mass selection, modified mass selection, year to row method, modified year to row method, then half shape selection, full shape selection, or modified half shape selection, or major one is recurrent selection. So this recurrent selection is again having simple types, simple recurrent selection, then recurrent selection for SCA, then recurrent selection for GCA, and reciprocal recurrent selection. So these are the some of the methods which we utilize in improving the cross-pollinated crops. So let us see first mass selection. The mass selection is the same as with the self-pollinated crops. The essentially from the maternal selection, since no pollination control is over there, and select desirable parents, then bulk the seed and repeat the cycle. This is the simple procedure of mass selection. And with the strict selection, breeder will reduce the population size and slow genetic gain since the lack of pollination control, and must be able to ID superior phenotypes. So not suitable for quantitative traits. This mass selection is not suitable for quantitative traits. Here is the overview of mass selection, in which the population grow the population and allow the random mating. There is no control over the pollination. Then they harvest and bulk seeds from the desirable plants, and then plant new generation and repeat this process. This is the simple procedure of mass selection. So this mass selection, higher percentage of the desirable genotype method can only be used in environments where traits is expressed and may not be suitable for off-season winter nurseries. The effectiveness is the function of heritability and manage field to enhance the differences. For example, irrigate excessively to increase the disease pressure. So these are the some of the merits and demerits of mass selection. So what are the achievements of this mass selection? The early varieties of bajra, that is Babapuri, Jamnagar, Giant, and Pusamuti. Then Toria increase the yield ability by 30% and oil by 56% in the Abohar varieties. Then in case of rye, many early erect and semi-erected type developed, that is type 11. Then yellow sarsa T2, T42 and T16, these are the examples. Then in case of brown sarsa, there is 17 dwarf, 17 medium, DS1 and DS2, these are the examples. Then in case of maize, T41, 19 and Jaunpuri. Then Desi cotton C402, and in case of Desi cotton again C520, and in case of American cotton 100F and 216F and castor B1 and B4. So these are the examples of the achievements by these using this mass selection. Next, there is some modification in mass selection called as modified mass selection, also known as stratified mass selection or a grid method. Stratified mass selection or grid method of mass selection. This modification is suggested by Gardner in 1961, and according to them, the field is divided into the several small plots. Example, having 40 to 50 plants in each plot, and selection is done within the plots and not among the plots. So this is the modified mass method, in which the different field is divided into the small plots and selection is done within that plot and not among the plots, and that's why the effectiveness of this mass selection is increased and hence called as modified mass selection. And seeds are selected and composite to the next generation. So this is the same like mass selection, but the one measure is taken that the field is divided so that the improve in the selection. The next is progeny selection, or also called as year to row method. Hopkins in 1908 developed this method by using or by extensively using maize. So what is this year to row method? A method in which selection is carried out on progenies derived from individuals' years is called as year to row method. So this is the definition of this progeny selection or year to row method. So what are the merits of this year to row method? It is based on the progeny test and not on the phenotypes of the individual plants. Hence it is a more accurate reflection of the genotype than the phenotypes. So the beauty of this year to row selection is that it is based on the progeny test, and that's why it is accurate than other method. Inbreeding may be avoided if the care is taken to select a sufficiently large number of plant population, and selection scheme is relatively simple and easy. What are the demerits? There is no control on the pollination and plants are allowed to open pollinate. Thus selection is based on the maternal parent only. So this is the major drawback of the year to row method. Then selection time is 2 years. Thus the time required for the selection is much as in the case of mass selection. Then there is also modification of year to row method. What is this modification is? Modification, the plant selected on the basis of the phenotype of open pollinated seeds from the each plant harvested separately. We can see in first year the plants are selected on the basis of phenotype, and open pollinated seed from the each plant is harvested separately. Then in second year the small progeny rows grown from the selected plants and superior progenies are identified, and the remaining seeds from the selected plants in the first year produce superior progenies bulk to raise the next generation. We can see here some of the seeds are grown and progenies are grown from the selected plants, and some of the seeds are tested for the progeny test, and among these plant producing superior progenies of that test are only selected from that generation, and these progenies are again in third year planted. The bulk seeds of the second year, that is item third, remaining seeds from the plants is planted which having superior progenies, and plants allowed to open pollinate, and plants with the superior phenotype selected and seed harvested separately, just like a second year only. And this is the cycle wise, this is the first cycle, this is the second cycle, and until and unless we achieve our goal we can continue this, and after that there is a preliminary yield trial, multi-location yield trial, and then release as a variety. So this is the modified year to row method in which progeny test is carried out here, and on the basis of that progeny test the progeny is again transferred to the next generation which is superior one. So what is next is recurrent selection. So what is this recurrent selection? The idea of the recurrent selection was independently given by Haber and Hayes and Garber in 1919 and East and Jones in 1920. So these are the two scientists in each group. They independently gave the idea of recurrent selection, but the term recurrent selection was coined by scientist Hull in 1945. So what is this recurrent selection means? The recurrent selection, as its name indicates, is defined as reselection generation after generation with the intermating of the selected plant to produce the population for the next cycle of the selection. So as name indicates, the recurrent selection is the reselection generation after generation with the intermating of that particular selected plants to produce a population and for the next cycle of selection. So it is a continuous process in which the superior polygenic traits are improved. The idea of this method ensures the isolation of superior inbreeds from the population subject to the recurrent selection. So there are four types of recurrent selection. So this recurrent selection is divided into four types. First is simple recurrent selection, then second is recurrent selection for GCA, third is recurrent selection for SCA, and last is reciprocal recurrent selection. So let us see first simple recurrent selection. So in case of simple recurrent selection, the several phenotypically superior plants are selected. So here is the population out of which these several phenotypically superior plants are selected, and these selected plants are self-pollinated. Here is the difference, that the selected plants are self-pollinated and self-seeds harvested separately, and self-seeds are evaluated and superior seeds are retained. And then in second year the individual plant progeny is planted from that selected seeds and all possible intercrosses are made. So here is the difference that the superior seeds or superior phenotypically plants are self-pollinated and they are harvested separately, and after that the self-seed are evaluated, and out of that the superior seeds are retained, and in next generation or in next year the all plant progenies are planted and all possible intercross are made there, and equal amount of seed from all the intercrosses are composite. So this is the original cycle in which the best progenies are selected, self-pollinated, then evaluated, and best seeds are retained, and in next generation they are planted and making intercrosses, all possible intercrosses, and after that the equal amount of seed from the each intercross is composited. So this is the original cycle, and next the again same the population is then planted and several phenotypically superior plants as like the first year or second year. The next recurrent cycle is continue, and this may be continue or repeated. Likewise the first recurrent cycle up to the desired results or desired improvements is observed. Then next type is the recurrent selection for general combining ability. Here is the procedure for the recurrent selection for general combining ability. So in case of general combining ability, it is a modification in the simple recurrent selection in which several phenotypically superior plants are selected as in case of the simple recurrent selection, and selected plants are self-pollinated and test cross as male to the number of randomly selected plants from the tester with the broad genetic base. So in case while we are testing the general combining ability, we use the broad genetic base tester, that is open pollinated variety as a tester, and we take here a test cross of the half of the selected progenies with this the tester, and test cross seeds are self seed from the each selected plants are harvested separately, and replicated yield trials are conducted using this test cross, and superior progenies are identified, and on that basis the self seed from the plants producing the superior test cross progeny planted in the crossing block. So some of the seeds are planted in the crossing block and some of the seeds are test cross. So on the basis of the seed from the plant producing the superior test cross progenies planted in the crossing block, all possible intercross are made between them and equal amount of the seed from all intercrosses are composited. So here again the same as simple recurrent selection, but here the case is different that the selected progenies or selected plants, superior plants in the original population, some of them are self-pollinated and some of them are test cross with this tester or open pollinated variety which is having broad genetic base. That's why we are testing here the general combining ability of that particular plant, and if it is found suitable then and only that the intercrosses are made between that particular plants and the seed is then composited. This is the original cycle. Then again first recurrent cycle just like a simple recurrent cycle, or again the likewise the original cycle, the second cycle may be repeated and so on, and this is again for the improvement in the general combining ability of a selected plant. So this is the general scheme or procedure for recurrent selection for general combining ability. The next is the recurrent selection for specific combining ability. So the breeding scheme for RS-GCA and RS-SA is similar, except that in case of open pollinated variety as a tester in GCA, in case of SCA, in case of reciprocal selection for specific combining ability, the inbreed is utilized and rest of all the process is similar. So in case of recurrent selection for the specific combining ability, only instead of this open pollinated variety as a tester, inbreed is utilized as a tester, and this inbreed is having narrow genetic base, because we have to test the specific combining ability of a plant with the particular inbreed. That's why here the narrow genetic based tester, that is inbreed, is utilized. Rest of all the scheme is similar. So the tester used in the recurrent selection for GCA is heterozygous or having broad genetic base, likewise open pollinated variety. But in case of SCA, it is homozygous or inbreed, and which is more effective with the complete and or dominance, and used when the additive non-additive gene action is important, and it improves the specific combining ability of the character. Then last one is reciprocal recurrent selection, RRS, a form of recurrent selection used to improve both GCA and SCA of the population of the character using two heterozygous testers. So here at a time two heterozygous testers are used. If we recall the earlier scheme, that in GCA we use the broad tester, in SCA we use the narrow tester. That means in SCA we utilize the homozygous tester, and in GCA we utilize the heterozygous tester as a tester, that is open pollinated variety, and in case of SCA we utilize the inbreed. But in case of reciprocal recurrent selection, at a time we use two heterozygous tester, and thus it is called as reciprocal recurrent selection, and it is also known as recurrent reciprocal half-shift selection, and it is given by Comstock and co-workers 1949. So main features of these methods are: it is used for improvement of the polygenic character, that is quantitative characters. Selection is made on the basis of the test cross performance, and two heterozygous testers are used as a source of population. Then it is used for improving the population GCA and SCA for specific characters. Then it is usually effective with the incomplete, complete and overdominance cases. It is used for improvement of those characters which are governed by both additive and non-additive genes. So, as we see earlier, the improvement of character which are governed by the additive genes, RSGCA is helpful, and while it is governed by non-additive gene, RSSCA is helpful. So in case of this reciprocal recurrent selection, it is used for improvement of the both character which are governed by additive as well as non-additive gene action. So this method is also require 3 seasons for completion of each cycle of the selection. Let us see here there are two population A and B. This A population is served as a tester for B, and this B population is served as a tester for A. And the test cross progeny of this B and A are taken here as A, and A and B as a B here. So these two populations are the source of tester for each other. Let us do the procedure. Several superior plants are selected in population A and B. So in each population, some of the superior plants are selected on the basis of phenotype. Selected plants are self-pollinated, and each selected plant from the A is test crossed with B with the several random plants from B, and the vice versa. That means the selected plants from the A is test crossed with the several random plants of the B, and reverse to that, the several each selected from plants from B is test crossed with the several random plants from the A, and this test cross and self seed from the each selected plants are harvested separately, and separate yield trials conducting the test crops progenies of the selected plants from the population A and B are maintained, and superior plants are identified. On that basis, the self seed from the, we can see here, the self seed from the plants that produce superior test cross progenies are planted separately for the population. So for population A, the superior self seeds are having superior test cross progenies or superior identified test cross progenies are planted here, and from B, the superior plants are planted here. All possible intercrosses are made among the progenies of A and separately that of progenies of B, and seeds from all intercrosses of the population A are mixed together, and those of B are also mixed together. Then composite these seeds from the intercross planted separately for population A and B, and next generation is growing, and here the next cycle is started. We can see here from the these two population, by using test cross progenies, superior plants are selected, and that particular superior plants are grown here. Intercrosses are made, and seed from A and B is separately composited, and from that the next progenies or next generation progenies or next cycle is started, and it is again grow as a original material, and next cycle is begin. So this is the original cycle, and from here the next cycle is started. So this is the recurrent reciprocal selection, in which number of cycles are depend upon the our aim, and here the improvement of both the GCA and SCA is simultaneously takes place. So what are the merits of that recurrent selection? Recurrent selection is efficient breeding method for increasing the frequency of superior genes for the various economics characters, and it helps in breaking the repulsion phase of the linkage, and it helps in maintaining the high genetic variability due to the repeated intermating of the heterozygous population. What are the demerits? It is not directly used for developing the new varieties. It is only used for the increasing frequencies of superior gene of various economic character in the variety. Then this method involves lot of selection, crossing and selfing work, and tedious one and laborious one. It permits selfing, which leads to the loss of genetic variability. So these are the some of the demerits of the uh, recurrent selection. So thank you. This video is for educational purpose only, and references are as below. Thanks once again.